Okay, can somebody confirm you can hear me? Anyone?
We can
We
hear
can
you.
I hear you.
All right. Before I begin, are there any questions? You share my screen here. Turn that down. All right. Not much to see there, but let's get rid of that. So let me remind you, today is October 28th. There will be a lab tomorrow, a lab quiz, not a lab quiz, a quiz tomorrow, quiz 3, covering labs 5 and 6, chapter 4, and the end of chapter 3. Today's lab is lab 7, throat and urine cultures. And let me remind you, I will be sending you your unknown number and your first 3 test results sometime tonight. It'll probably be late tonight. Any questions about anything? All right, let's move on. I think I can shut this down. Lab 7, throat and urine cultures. There's a little bit to read. A page on nose, throat, and mouth normal microbiota. You can read about beta hemolytic streptococci, Haemophilus, and the coagulase production, all in the appendix of this lab module. So just scroll to the end of the lab module, you get an appendix, and I put in these 3 items. You need to read about the oxidase test on page 132 and the coagulase test on page 155. So not much reading to this week. Supplemental materials, we have some video clips in this lab. Oops. The learning objectives: 1, be able to explain how to properly collect throat and urine specimens for routine laboratory culture. And 2, list common bacterial pathogens isolated from throat and urine cultures. 3, know the culture media used to cultivate these pathogens. 4, recognize which tests are used to identify throat and urine pathogens. Sorry, I'm trying to look for something I need to tell you about the unknown project, and I'm not finding it. Hopefully I'll find it, but 5, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. So you do need to understand those. And be able to explain them. We're going to talk briefly about the XV factor testing, or the 10-5 vector testing. I think it's XV. But you do not need to understand them or explain them. Any questions about any of that? All right, I have 2 things to tell you about the unknown project that I forgot to tell you when I was discussing the unknown project about 2 labs ago, and that is the unknown notes pages. It is going to be graded. The unknown notes pages will be 5 points, which will count the same as a normal lab. So the unknown notes pages will be graded. So make sure you do good job on the unknown notes pages. And then the size of the report should be between 4 and 6 pages long. I'm talking about the body of your text. I'm not talking about the bibliography or any pictures you add, but the size of your text should be 4 to 6 pages long. If it's outside that range, there will be a penalty. And this is discussed in your unknown project instructions, but I forgot to mention it. So I'm mentioning it now. Any question about any of that? One student asked me, what if it's 3.5 pages, meaning the fourth page is not complete? And I said, that's 3.5 pages, you'll get a penalty. If you have more than 6 pages, you'll also get a penalty. That rarely happens, but um, I don't want to report that's 10 pages long because I have to read it. So 4 to 6 pages, ideally around 5, but I'll let 4 to 6, no penalty. Beyond that, you'll get a penalty. Any questions? All right. So let me cross these out. I've talked about them now. Sorry about missing them when I was talking about the unknown project. Introduction. Once specimens are received in the lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for each um, specimen coming into the lab for the throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For throat cultures, we only have two culture media. Blood agar, the main one, and then chocolate agar. The main bacterial pathogens that's isolated in the throat is Streptococcus pyogenes, and it grows on blood agar. It is fastidious, but blood supplies the nutrients that it requires to grow. Chocolate agar would work, by the way, but blood agar is what's typically used. And then the other pathogen is Haemophilus influenzae, which is common in babies and young children. It is not so common in adults or even older children, but it is a common pathogen in babies and very young children. And this is a fastidious organism, but it cannot grow on blood agar. And that's because Haemophilus influenzae cannot lyse the red blood cells. And so the ingredients in the blood do not get out to Haemophilus influenzae for it to grow. But you can grow it on chocolate agar because chocolate agar is the same as blood agar. It's just that you add the red blood cells when the agar is immediately taken out of the autoclave, meaning it's very, very hot, and that causes the blood cells to lyse, releasing the additives in the blood that Haemophilus influenzae require to grow. And it also gives the blood the chocolate agar of brown color, which is why it's called chocolate agar. Any question about throat specimens? All right, urine specimens include a number of different species over here. We tend to grow urine specimens always on two media. We always put it on blood agar, but we also put it on McConkey's agar or EMB. So we always use blood, and then you always use either McConkey's or EMB, one of the others. The reason for a blood agar is it can grow all cells, but and particularly the gram positives, which Streptococcus and Enterococcus and Staphylococcus can be a urinary specimen causing a urinary tract infection or disease of it's in the urine, and it can grow on blood agar. But most of the urinary tract infections are caused by gram negatives, especially from the family Enterobacteriaceae, with the number one urinary tract infective agent being E. coli. These are all gram negatives, and that's why we put it on McConkey's or EMB agar to get the gram negatives to grow and then to remove the gram positives. One problem with Enterobacteriaceae, it is gram-negative, but it's very similar in shape and most appearances to Pseudomonas. And there is some species of Pseudomonas, particularly Pseudomonas aeruginosa, which can be a urinary tract infective agent. Any questions about what media we grow these specimens on? All right. For throat cultures, a sore throat may be caused by viruses or bacteria. If the sore throat has redness and white spots, particularly among the back of the throat and the tonsils, the white spots are indicative of a bacterial infection. The redness, if it's only redness, is indicative of a viral infection. In this lab, we're only going to talk about bacterial infective agents. Shrink that down a little. The most common isolated bacterial pathogen from the throat is Streptococcus pyogenes. That's its scientific name. It's also called Group A Streptococcus, or GAS as an abbreviation, G for Group A for Group A, and then S for Streptococcus. Strep throat may be diagnosed in one of two ways, the rapid testing or the throat culturing. We're only going to talk about the throat culturing. You swab the back of the throat where the tonsils are or where there's redness or white patches, and you do not touch the tongue. You do not touch the sides of the mouth, the teeth, or the top of the mouth. A tongue suppressor helps to hold the tongue in place and get the swab to the back of the throat. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy, which is why it's important to test for Streptococcus pyogenes, because Streptococcus pyogenes has a cell capsule and it resists the host immune system. So many patients, when they have Streptococcus pyogenes, they cannot throw off Streptococcus pyogenes. And that's why you put them on antibiotic therapy. Now, in the old days, all they did was they simply cultured the throat on blood agar and then looked to see if there was a small white colony. Let me blow this up. With a zone of hemolysis around it. So right there, you can see a very small white colony with a very large, clear region of hemolysis. There's another small white colony with a very large region of beta hemolysis around it. In the old days, that's how they did to test for Streptococcus pyogenes. And we now know, and I don't know if they knew that back when I was a child, but they now know that there's other bacteria that can cause a similar reaction like that, meaning other members of the family Streptococcus. And so you need to do the blood agar swipe and then see beta hemolysis, and then do 2 additional tests to prove that it is Streptococcus pyogenes, too small. You do the Cadlase test and the bacitracin susceptibility test. So you get the bacteria having beta hemolysis on a blood agar plate and put it on another plate. And then you apply Cadlase or hydrogen peroxide to the cells to see if they produce Cadlase. Hydrogen peroxide in the presence of Cadlase makes water or hydrogen peroxide convert into water and oxygen. The oxygen produces bubbling in the hydrogen peroxide. And If the organism produces catalase, it is not a member of Streptococcus. It might be Staphylococcus or something like that. Most organisms that grow in the air produce catalase, but there are two organisms that grow in the air that do not, meaning two families, and that's family Streptococcus and family Enterococcus. They're negative for catalase production. And let me see if I can move that up now. Nope, that should be moved down. That one should be moved up. For some reason, these got moved and it's not showing now. I have to go through and change this. This page I've been working on and I need more time for it. Oh, I got to show you the, I got to show you the, well, I might not be able to show it to you because I'm not seeing the, hmm, I don't see how to move that to the background. Um, Let me look for another copy so I can show you the picture. I moved it up and then it went to the background for some reason, and that's why you can't see it. All right, it's not in the right place here either. Oh, there it is there. So you put the bacteria in a drop of hydrogen peroxide, and if it's negative, no, meaning no bubbles in the hydrogen peroxide, this is indicative of the family Streptococcus or Enterococcus because they're the two main families of bacteria that are catalase negative and yet grow in the air. Most bacteria are catalase positive if they grow in the air, and then you'll see bubbling in the hydrogen peroxide like that. Any question about any of that? All right. And then the second test you use is the base trace and sensitivity test. You take the bacteria from the blood agar and then streak it in this region of the plate here, put on a disc containing the antibiotic bacitracin in it, allow the bacteria to grow, incubate, and if you see a zone of inhibition around the disc, as shown here, then the test is positive because the bacteria are sensitive to bacitracin, and group A Streptococcus or Streptococcus pyogenes is sensitive to bacitracin. If, on the other hand, the bacteria is resistant to bacitracin, then the bacteria is not Streptococcus pyogenes. Any questions about this bacitracin test and how it shows that you have, well, Streptococcus pyogenes, because this is the only one that is. That's catalase negative, and then is beta hemolytic on blood agar. Any question about any of that? So you really need two additional tests after you get beta hemolysis on blood agar to show that it is Streptococcus pyogenes. Go back to my other one. There we are. Another organism which is found in the throats, particularly in infants or very young children, is Haemophilus influenzae. Haemophilus influenzae is a gram-negative coccobacillus, and it's common cause of eye, ear, and sinus infection in babies and young children. Not so much in older children and in adults. Haemophilus influenzae is fastidious, a tiny gram-negative coccobacillus, requiring the growth factors NAD and hemin, and that's the 5 factor and the X factor, which are supplied in chocolate agar. It is in the red blood cells of blood, but Haemophilus influenzae cannot lyse the red blood cells to get the growth factors that it needs. And so it will grow on chocolate agar, but not on blood agar. Let me show you that right there. So we streaked Haemophilus influenzae in this region here, and you see in this whole region there is no growth of Haemophilus influenzae because it needs NAD and hemin, which are found in red blood cells, but it cannot lyse the red blood cells to get access to the NAD and hemin. However, if you were to streak a streak of Staphylococcus aureus down the blood cells like that, let me blow this up, and then streak the Haemophilus influenzae from here to there, you'll note that Haemophilus influenzae grows only in this region here around the streak of Staphylococcus aureus. Why it's growing here is because the Staphylococcus aureus can lyse the red blood cells, releasing the additives from the blood that Haemophilus requires, meaning NAD and hemin. And so the Haemophilus influenzae can grow in this region, but not in this region. And I don't know if that's streaked down here, not in that region either. Only around Staphylococcus aureus. Any question about that? All right, so that's it with the throat cultures. Let's talk about urine cultures. Urine cultures are used to diagnose urinary tract infections, UTIs. Bacteria can travel up the bladder through the urethra. Females are more predisposed to developing a UTI than males simply because females have a shorter urethra than males. There are 3 ways you can sample the urine, or I should say 3 recommended ways you can sample the urine. The first is the clean catch midstream method, where the patient cleans their genitals prior to urinating, and then they start urinating, and they catch the urine in the middle of the stream, not at the beginning of the stream, not at the end of the stream. If you collect the urine in the middle of the stream, you'll have less contamination from the urethra than if you catch it at the beginning of the urine stream or at the end of the urine stream. Any question about that? And if you don't know, the urethra already is colonized by microbes. So there's a normal microbiota on the urethra. And if you're collecting in this fashion, you will pick them up. There's no way to avoid it in the clean catch midstream, but you can reduce their number. Another way, which patients don't like because it's somewhat invasive, is sticking a catheter up the urethra into the bladder and then sampling the urine in the bladder. However, the catheter is touching the urethra. It can pick up bacteria and then put it into the bladder and then sample the bacteria that way, meaning pulling up the contamination from the urethra. So the catheter is better, but it is not, I don't know, error prone. It's not foolproof. You still get contamination. The best way to do it is the suprapubic aspiration, where you put a needle through the abdominal wall. Obviously, you sterilize, not sterilize, but alcohol swab the abdominal wall, put the needle in, put the needle into the bladder, and then sample the urine directly from the bladder. And that will be much less contamination. You totally avoid the urethra and the lots of contamination. However, you should know that, at least in females, the bladder does have some normal microbiota in it. And I don't know the question of whether males have normal microbiota in their bladder or not, because I've never found anyone who studied males to find out if their bladder has normal microbiota or not. They might not, because the longer urethra prevents the microbes from getting into the bladder. But then again, they might, nobody's ever studied it, and so I just don't know. Any questions about the 3 ways you can collect a urinary sample? Understandably, patients much prefer the clean catch method, and they least prefer the suprapubic aspiration method. All right, once the specimen is received in the lab, 0.001 milliliters, that's 1/1000th of a milliliter, or simply 1 microliter, of urine is put on a blood auger plate. You normally streak it this way and then streak it with an S-type movement, so you get something like that. And then you also take this urine and then streak it on a McConkey's plate or an EMB plate, and that's to grow gram negatives. You will get gram negatives growing on the BAP, blood auger plate, but you'll also get gram positives. On the McConkey's and the EMB, you'll only get gram negatives. Coliforms such as E. coli are the most common isolate from a urine specimen, especially E. coli is the number 1 possibility, but the members of the family Enterobacteriaceae are very commonly found in urine specimens. The interpretation of a positive urine culture, because you'll always get a positive culture unless you're doing suprapubic aspiration on a male, maybe in the bladder. You should always get positive results, because you'll pick up contamination from the urine. And then even in suprapubic aspiration in a female, you will pick up normal microbiota that are present in the bladder. So you almost always get a positive urine culture. And that's a good question. If the males underdo suprapubic aspiration, if they have a normal microbiota, it should show up. So I should look into that and see if anyone's done that, because that would be evidence sort, but I haven't done that. Anyways, the positive urine culture is multifaceted. Factors that need to be considered are the type of collection. Clean catch midstream will be much more contaminated than catheterized specimen, which would be much more contaminated than suprapubic aspiration. The gender of the patient has to be considered. Males have much less contamination than females. I'm not sure why that's the case, but that is commonly seen. And then the number of colonies per mil of urine is something to consider. And the presence of polymicrobial growth. If there's multiple organisms isolated, it may indicate poor collection technique and a lot of contamination from the urethra. Males normally get 50,000 colonies per mil of urine, Females normally 100,000 colonies per mil. I assume this is of the voided urine. All right, once you get your bacteria up and growing, you then need to run some tests on it. The first test we're going to talk about is the coagulase test. Coagulase is looking for the production of the enzyme coagulase, which is used to differentiate Staphylococcus aureus from other members of the Staphylococcus family. The other members of the Staphylococcus family are all coagulase negative. Staphylococcus aureus is coagulase positive. Staphylococcus aureus is coagulase positive, and so it can convert blood plasma proteins, fibrinogen, into fibrin, resulting in a blood clot. So in plasma or blood or serum, you have, you have fibrinogen and coagulase. If that enzyme is around, it can change the fibrinogen into a fibrin clot, which causes a clotting of the blood or the serum. There's two tests you can use. The rapid slide coagulase test, where you put plasma on the slide, you put the bacteria in it. If the bacteria lack the coagulase enzyme, then the smear will be uniform and cloud-like. I don't know what you call that, creamy-like. If it does produce coagulase, the cells as well as the serum or plasma will begin to clump, forming this type of a pattern. And that's from the small clots or clumping. You can also do the tube test, where you have the plasma in the tube, which is a liquid, and then you put in the bacteria. If the bacteria produce the enzyme coagulase, you get a clot, which is shown in the first slide here, the positive. And if it's coagulase negative, the tube will continue to be liquid. Any question about any of that? So positive and positive, negative and negative. All right. Remember, the most common bacteria isolated from a routine urine culture is E. coli. And then the other most common are members of the family Enterobacteriaceae. But there are members of the Pseudomonaceae family, especially Pseudomonas aeruginosa, which can cause a urinary tract infection. How most people differentiate the members of Pseudomonaceae from the members of Enterobacteriaceae is with the oxidase test, because both members, most families, are gram-negative bacilli, but the Pseudomonaceae family is oxidase positive, all members, and the Enterobacteriaceae family, all members are oxidase negative. There are some other differences between the family. The family Enterobacteriaceae can all ferment the sugar glucose. The family Pseudomonaceae, none of the members can ferment the family glucose. There's one other difference. What the heck is it? They're both gram negatives. Oh, yeah. The family Enterobacteriaceae, all members are facultative anaerobes. The members of Pseudomonaceae, all members are aerobes. So those are three ways you can tell the families apart. We usually just do the oxidase test because it's simple and easy to run. You get the results, well, 30 seconds after you put the reagent and the cells together. So very quickly you get the results. But you could do the glucose test. You'd get that result in 18 to 24 hours. And you could do the um, looking for the growth in the organisms, whether it's a facultative anaerobe or an aerobe, and that would take 2 days to do. And we talked about that a little bit, the thioglycolate too. All right, any questions about any of that? Um, so the oxidase test looks at the enzyme cytochrome C. Cytochrome C oxidase is its long name, which is a member of the electron transport chain. So this organism, if it's Pseudomonaceae, can perform aerobic respiration, having cytochrome C oxidase. And then you just put the cells together with the reagent and you look for a indigo or purple color happening within 30 seconds. And you have your test results. If it's negative, then it's negative for the oxidase test, meaning no color change. If it turns purple or indigo, it's oxidase positive. You must do the test within about 30 seconds, meaning you don't let this reagent sit for 2 minutes or longer, because there's a side reaction going on, which does take longer, and it can get the test to appear purple, and that's why you let the test run for only about 30 seconds. I think specifically it's 20 to 30 seconds. And longer than that, you say it's oxidase negative. All right. Let me step back. I forgot to talk about Staphylococcus aureus and why, if it produces coagulase, why that's potentially a problem for the patient. Where the heck is that? Here it is. Um, Staphylococcus aureus produces the enzyme coagulase, which causes a blood clot to surround the Staphylococcus aureus when it's first infecting the blood or the patient, when the bacteria numbers are low in numbers. When the bacteria are low in numbers, it's easiest for the immune system to remove the pathogen. And coagulase helps protect Staphylococcus by creating a blood clot around the bacteria. That makes it much harder for the bacteria to mount an immune response or phagocytize the Staphylococcus aureus. Curiously, when the Staphylococcus aureus has grown and is large in number, and it needs more space than the blood clot is providing, the Staphylococcus aureus digests the clot, and then the Staphylococcus aureus can spread in the blood, and the host immune system tends to be overwhelmed by the large number of bacteria in the blood. Meaning now the health immune system can fight Staphylococcus aureus, but it's ineffective because there's so much Staphylococcus aureus, the host immune system is overwhelmed. And that's a little bit about Staphylococcus and why it's a dangerous species if you get it. It has, well, various pathogenic factors, one of them being coagulase, which tends to make the bacteria more severe in the diseases it causes. Any question about any of that? All right, need to talk about the summary here. So urine and throat cultures are two of the more common types of specimens received in the lab for bacterial cultures, and microbiologists are trained to isolate and identify pathogens present in both. You can take a look at the references. We're going to be doing an online class, so there's not a whole lot to do, but you will be not physically performing any of these activities, but you will be responsible for knowing how to perform them and interpret them. We have a little video on the catalase test, so watch that video. We have a little video on the coagulase test, so watch that video. And we have a little video on the oxidase test right here. All right, let's go to the questions in the worksheet. Come on. Which I think I've changed the questions a little bit from my copy there, but this should be your questions. So question 1A, refers to, 1, 2, 3, and 4 refer to the enzymes catalase, coagulase, and oxidase. 1A, what enzyme converts fibrinogen into fibrin? And if you give me an enzyme you get from AI and it's not discussed in the lab module, it is wrong. Okay, so you should be getting your answers from the lab module. Meaning if it's something we didn't talk about, the answer is wrong. Name a microbe that tests positive for this enzyme. Here you can probably use AI. I'm not going to mark you down there, but there was one mentioned in the lab module. And 2A, which enzyme detects the presence of cytochrome C proteins in the electron transport chain? And then 2B, name 2 microbes that test negative for this enzyme. You can give me the family names here or 2 species. I think the lab module gave you 2 families. 3A, which enzyme hydrolyzes hydrogen peroxide into the non-toxic products of water and oxygen? And 3B, name 2 microbes that test negative for this enzyme. Once again, I think you can name families or you can name species. 4, which enzyme is used to differentiate between the following? So which enzyme differentiates between Staphylococcus and Streptococcus? Which enzyme differentiates between Staphylococcus aureus from other members of the Staphylococcus family? And which enzyme can distinguish between Enterobacteriaceae, like E. coli, and Pseudomonaceae, like Pseudomonas aeruginosa? 5, concerning specimen collection for routine throat culture, what areas of the throat should be swabbed? And I changed that to plural, so you must give 2 answers there. And then what areas of the mouth should be avoided? And you should also give at least 2 answers there. 6, what is the most common species causing bacterial throat infections? 7, what test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus, meaning Streptococcus pyogenes, from other beta hemolytic Streptococcus species isolated in a throat culture? And note, this is a little different than the lab module because we already are given it's beta hemolytic and that it is a member of the Streptococcus family. And that's why you only need 1 more test to distinguish between Streptococcus pyogenes and another beta hemolytic Streptococcus species. 8, answer the following concerning a child's throat culture. State 2 non-selective agar plates that are inoculated. And 8B, what organism can be isolated on CAP but not on BAP? CAP standing for chocolate agar, BAP for blood agar. And then the P is for plate. 9, list 2 media that are used to grow urine cultures. It must be one of the 3 mentioned in the lab module. 10, list 3 reasons why urine specimens should be rejected for culture. You can get that out of the appendix, meaning I added that to the appendix. 11, how much urine is placed on the surface of a blood agar plate? 12, what is the most common bacteria isolated from patients with a bladder infection. 13. Identify the Gram stain, the Gram reaction, and the cell morphology, the shape of the bacteria, not the cell growth arrangement. If you can't find an answer in this module, which I think it is, all of them are in here, you may need to do some research. Like Haemophilus influenzae, you can find in the lab appendix. And then 13A, what's the Gram reaction and the cell morphology of Pseudomonas aeruginosa? B, the Gram reaction and the cell morphology of E. coli. C, Staphylococcus aureus. D, Streptococcus pyogenes. And E, Haemophilus influenzae. Any questions about this lab? All right, I will be here until 7:45. The last student leaves to help you with any questions. So get started with the lab. Remember, if you have a question, to ask before the last student logs out, because once the last student is logged out, I'm going to log out as well.